TGI Monday. Good morning, everybody. How are you guys? So, I'm going to go over the second part of this practice test, but before I do that, I want to talk to you guys about test preparation. So, let's say we make a spectrum right here. So, here's our spectrum. And on this side of the spectrum represents when you know nothing concerning preparation for a test, a history test, a math test, a biotechnology test, whatever. And over here, you know everything. So this is something that's going to affect your performance on a test, how much you know. But that's not all that's going to affect your performance on the test. So we could look at this spectrum. We could use it to look at another spectrum, which is how well you do on the test. So over here, you get 0, the test kills you. Over here, you get 100, you kill the test. So if we use this green dot right here to symbolize how much you guys know for this BACE biotechnology test, I'd say you guys are right about here. You guys know a lot, but there's things that are going to be on the test that I haven't covered, and that's just how it is. So this, let's say, is where you're at. And of course, different people in the class are different places. There's some people over here and there's some people over here, right? Well, what I'm trying to tell you is you can rig it so that you perform better than you're prepared to perform. And these are the two things that I'm here to teach you guys by going over these tests. I'm here to teach you content, but I'm also here to help you perform on the test better than you should perform based on how much stuff you know. And this is your key to success in college. Your key to success in college is to develop skills so that you perform better on tests than you should. All righty. So let's go check out this test here. Okay. Question 1. What is a disadvantage to using prokaryotes in the production of biopharmaceuticals? So prokaryotes, those are bacteria, and pharmaceuticals means drugs or medicines. Biopharmaceuticals are drugs or medicines that are produced by cells. So when we trick bacteria into producing human insulin for us, we purify that insulin and package it and give it to patients. That's called a biopharmaceutical. So, which is a disadvantage to using prokaryotes in the production of biopharmaceuticals? Prokaryotic cell cultures are more susceptible to contamination than eukaryotic cell cultures. Well, both kinds of cell cultures are actually pretty susceptible to contamination. And eukaryotic cell cultures, that would be growing like, for example, hamster ovary cells or HeLa cells, they're totally susceptible to contamination. That's a crappy possibility. Prokaryotes are unable to properly fold and assemble more complicated eukaryotic proteins. So you guys know that prokaryotes do not do RNA processing. They skip straight from transcription to translation, whereas eukaryotes like us, we're able to do gnarlier stuff. We're able to make more complicated proteins. Like, for example, you can't get bacteria to produce hemoglobin. You can't do it. They're not that rad. Prokaryotes cannot accept mammalian DNA. We know this to be false because we can get prokaryotes to produce human insulin for us. So that's a crap answer. So crap answer. This answer seems the least crappy to me, so I'm going to pick it. Which structure regulates the flow of molecules into and out of the cell? Well, that would be the plasma membrane because that's the boundary between the inside and the outside of the cell. Why is E. coli usually incubated at butt temperature? Well, E. coli doesn't only grow at butt temperature, it just grows fastest at butt temperature. What's butt temperature? 37°C. What's butt temperature in Fahrenheit? 98.6 degrees Fahrenheit. Which of the following serves as a decolorizer in a gram stain? What? Gram stain decolorizer? What even? And here's another one. Okay, here's what even. Gram stain is a process of trying to stain bacteria, and they turn different colors based on what kind of bacteria they are. So it's a method to identify what kind of bacteria you're looking at. It's not really used anymore because now we have DNA technology. You can just sequence the bacteria and figure out what kind of bacteria it is, but they have it in the test because it's old school. I didn't teach it to you guys because it's old school, but now I guess I have to teach it. So here's the deal. You swab a patient, you swab a doorknob, you swab something, and you get all the bacteria off of it. There's different shapes of bacteria. There's rod kind of bacteria. There's cocci shaped bacteria. There's spiral shaped bacteria. So that's what this is attempting to show. You apply crystal violet, which is a stain. You apply iodine, which is a mordant, and mordant means that it makes the stain stick. And then everything turns purple. But then you use alcohol and you wash the cells. And any cells that didn't actually take in the crystal violet, the alcohol just washes it right off. But if cells took in the crystal violet, the alcohol does not wash it off. Then last, you add a different stain, a counter stain called safranin, which is pink. And that, because these cells would be invisible under the microscope if you didn't stain them some color. And the pink stuff won't show up in cells that are already colored purple. So then you can look, and the cells that took up the crystal violet are called gram positive. Positive means yes, they took it up. And the cells that didn't take it up are called gram negative. And the reason some cells take it up and some cells don't is because the structure of different bacterial cells is different. In gram negative bacteria, there's actually two plasma membranes sandwiching a layer of a molecule, a polymer called peptidoglycan. I don't know if I pronounced that right, but peptidoglycan. And in gram positive, that's purple staining cells, they have a huge thick layer of peptidoglycan. In fact, in gram positive bacteria, once you get all the water out of them, the weight of the cells is like 90% peptidoglycan. So there's a buttload of it in there. And gram negative cells also have peptidoglycan, but their dry weight mass of peptidoglycan is only 10%. So anyway, that's that. And I'll show you what this peptidoglycan looks like. It's kind of cool. It's a polymer. So the monomer unit of this polymer is right here. It's made of two sugar molecules hooked up to a short peptide. So 4 amino acids. So 1, 2, and then 1, 2, 3, 4. And you can see how the amino acids hook to each other and the sugars hook to each other. And it makes this polymer right here, which gives the cell structural shape. The reason it's important, this might come up in the test, the reason it's important is that cells that are gram positive are harder to kill with antiseptics. Antiseptics are things like bleach and alcohol. But gram positive cells are easier to kill with some antibiotics. Some antibiotics interrupt the synthesis of peptidoglycan and therefore prevent cells from reproducing. So some antibiotics only work on gram-positive bacteria. That's why it's important for physicians to identify what kind of bacteria you have an infection with, and that will inform their decision on what antibiotic to give you to kill those suckers. So anyway, which of the following serves as a decolorizer in a gram stain? So that's like a solvent, and alcohol is a solvent. Boom. Which of the following is an accurate description of gram-positive bacteria after gram staining? Well, after you gram stain gram-positive bacteria, they're going to be purple, and they're going to be purple because they have that thick-ass layer of peptidoglycan. So that answer's right here, purple, because they got a thick layer of peptidoglycan. Did you guys see that? It's that answer right there. Oh. Question 6. When exposed to a foreign antigen, remember an antigen is something that causes an immune response, like pollen or a germ. When exposed to a foreign antigen, the normal human immune system response is to produce which of the following? We're going to look for something that's involved in immune system functioning. Red blood cells? No. White blood cells? Yes. But red blood cells? No. They're involved in gas exchange. Hormones? No. Hemoglobin? No. Antibodies? Yes. 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 This is a possible right answer as we learn with that cytokine storm stuff, but boom, this is what we're looking for, antibodies. Okay, why are biopharmaceuticals, again, biopharmaceuticals are pharmaceuticals made by cells, so they're usually proteins actually. Why are biopharmaceuticals typically more difficult to manufacture than traditional chemically synthesized pharmaceuticals? Remember when we made the aspirin and you guys just mixed the stuff in the test tube? It's just crystals. The aspirin is just what's aspirin called? Acetyl salicylic acid. It's just crystals. You can like dissolve them, you can like precipitate them, you can heat them, you can autoclave them, whatever. They don't care. But you can't do that to a protein. Remember how we have to keep everything on ice all the time and keep it covered or we'll mess it up? It's because biological molecules are more sensitive. They're easier to mess up. So more prone to contamination? Maybe. Biopharmaceuticals cannot be heat sterilized? Yeah, that's right. Biopharmaceuticals typically require specially handling and storage? Yeah, that's right. Whenever I see two that I know are right and one that's like, I don't know, and they give me all the above, I'm going to click all the above. Booyah. Which of the following features of bacterial plasmids can be used as a marker when growing bacterial cultures? Marker, selectable markers. Oh yeah, those are those things that help us choose which cells got genetically transformed and which ones didn't get genetically transformed. So, multiple cloning site? No. That's a place in a plasmid that has a whole bunch of recognition sequences for restriction enzymes. No. Antibiotic resistance gene? Yes. That is what we use as a selectable marker. I'm just going to read the next two just to be sure. Arabinose operon, that's a way to turn gene on and off. And no, we don't use that as a selectable marker, although we can. So this question, I don't really like it that much because this can be used as a selectable marker. Origin or replication has nothing to do with it. This is the best answer, although yes, you could use an operon as a selectable marker, but antibiotic resistance genes are the go-to standard for selectable markers. That's the best answer. The observable traits of an organism are referred to as the phenotype, man. That's just what it is. It's an observable trait, bald phenotype. I have a genotype that leads to this phenotype right here. It's what you actually see from the gene expression. According to the central dogma of molecular biology, DNA is transcribed into RNA, which is translated into a protein. Central dogma, better know it. If you don't, you're gonna get destroyed. Let's see how we did. Think we got any right? Oh, we got them all right. All right, you guys. Happy Monday, you friends. I'll see you guys on Wednesday with a video. So tomorrow you're just going to do quiz number 3. Sound good? Today, that's it for today. I hope you guys took some sweet notes because you're going to want to use them. All that peptidoglycan stuff, go back and take notes on that if you didn't. I forgot to tell you.